let's see. Hi, Wolfgang, are you there?
Yes, I'm here.
Hi, great. Glad to see you again.
Hi.
Yeah, and now we have our keynote speaker, and I'm the one to coordinate the Dr. Liner's presentation. So before Dr. Liner's presentation, I like to give some introduction of Dr. Wolfgang W. Liner. So Dr. Liner is the chief of the innate immunity section of the basic immunology branch division of energy immunology and transplantation. And it's a National Institute of Energy and Infectious Diseases. So it's often we call it NIAID or NIAID. So he oversees the NIAID vaccine adjuvant program. And his section manages NIAID's grant portfolio in the area of innate immunity. Wolfgang joined NIAID's actual Mural program in 2008. And after working on conventional and self-replicating DNA and RNA-based vaccines and adjuvants for melanoma and T-cell lymphoma at the National Cancer Institute for a decade, he became interested in DNA vaccines during his postdoc at the Walter Reed Army Institute of Research, where he developed the first DNA-based vaccines for Plasmodium burgdorferi malaria. So Wolfgang received his PhD from the University of Würzburg, Austria, defining the immunological differences between the spleen and the splenic autotransplants, which are responsible for the impairment of certain immunological functions of the transplanted tissue. Dr. Liner is currently leading a program called VAC, it's a vaccine adjuvant portfolio in NIAID. And he has also contacted me about how we can work together and use vaccine ontology to standardize the adjuvants in the VAC program. So it's a very good opportunity for us to work with Dr. Liner, and I'm happy to invite Dr. Liner for the presentation here. Thank you for accepting our invitation. Okay, without further ado, go ahead. Yeah.
Thank you so much for the introduction. Thank you so much for the invitation. So my talk's going to be somewhat different from what you've heard so far, but you'll see hopefully in a little while why this is relevant for this conference. As Dr. Hayes said, I oversee a program at the NIAID to create new adjuvants and just to put things in context where I am. At the extramural program of NIAID, we have three scientific divisions for infectious diseases, for HIV, and then the division I am in, allergy, immunology, and transplantation. And among all the different programs that these divisions are involved in, two of them are, of course, very interested in vaccines for infectious diseases. The majority of infectious diseases by our infectious disease division, and then HIV by the division of AIDS. Our division has a large program to support the discovery and development of novel adjuvants. And these are meant to flow back into, flow back into the programs for novel vaccines, but also into our programs for vaccines against immune-mediated diseases, so allergy, asthma, tolerance induction after transplantation, autoimmunity. They're also supposed to become available for other government agencies but equally important, become available for vaccine developers outside of the NIH. So when you look at adjuvants, at the overall topic, what's the issue? From a regulatory standpoint, very unexpectedly, if you think about what adjuvants do, the only component of a vaccine that's considered to be the active ingredient is the antigen. In case of a subunit vaccine, it would just be the protein. Adjuvants are classified as an inactive ingredient, again, from a regulatory standpoint only. However, all vaccines do need adjuvants, and these could either be naturally occurring, so in case of an inactivated pathogen or a subunit vaccine, these are components that are already part of the pathogen. Or if you are cleaning up the vaccine, if you're only delivering just a protein, you have to exogenously add the adjuvant. And traditionally, that's been an aluminum salt. And these aluminum salts have been used in humans for now over 100 years. They're safe. They've been used in literally hundreds of billions of doses, but they're pretty weak. And for a number of diseases, they're just not cutting it. They're not powerful enough. They're also not inducing the right immune profile. So when you look at different types of immune profiles that are induced, alum drives one specific type of immune profile that just isn't useful for certain diseases. Unfortunately, when you look at licensed vaccines, there aren't a lot of alternatives. Only 5 of the FDA-approved vaccines are based on novel adjuvants. Now, why is that? It is pretty difficult for vaccine developers to access any of those late-stage adjuvants, difficult or impossible. To develop your own adjuvants is expensive. It's risky because they may not work, they may not be safe enough, and it takes a really long time. So this is what our program is trying to address. We're trying to bridge that gap between the adjuvant research community and the vaccine research community and make novel, safe, effective adjuvants available for novel vaccines. I'm not going to talk about all the different programs we have to support adjuvants. I just wanted to give you that overview and point out that in the course of developing an adjuvant, you're looking at these different stages. So initial discovery, mechanistic types of studies, looking at combination adjuvants and why certain combinations are synergistic. Then there's this long valley of death when it comes to product development, where you are doing a lot of studies to establish the usefulness, safety of a novel adjuvant. That's the adjuvant development program, and then eventually taking them into clinical trials. So for each of those stages, we have different mechanisms, different solicited programs to support the discovery and development of novel adjuvants. And it's not just against infectious diseases. You will note here, we have a program for developing novel adjuvants for substance abuse vaccines, so specifically opioid vaccines. Now, because of the title of this workshop focusing on vaccines and drugs, I wanted to point out the following. When you look at adjuvants, this is really a topic where vaccines and drugs intersect. So traditionally, when you think about an adjuvant, you don't think about drugs. You think about these complex molecules that have been discovered by chance. Traditionally, they are natural products. They're purified from some natural source and maybe modified afterwards. But that means they all depend on natural sources. And especially in the context of the current pandemic, if you think about the next vaccine in line for approval, the Novavax vaccine is based on an adjuvant that relies on a tree bark as a source of that adjuvant. This is a tree that grows in one area, in one country. It's a limited resource. Another one, the one that's being developed by GSK and Sanofi, relies on an adjuvant that is based on shark liver oil. So once again, a natural resource, very limited. These traditional adjuvants, because they're often complex, they're often not pure molecules. And as our expectations of the quality and purity of products being injected into people keeps increasing, that does increasingly become a concern. Now, when you look at modern adjuvants, the approach is very different, and what they look like is very different. They're discovered through systematic high throughput screens, targeted screens, where you know the target molecule, you know the target receptor, and then look for agonists of that receptor. They are synthetic molecules, which means they're highly pure. It's also easier to manufacture, and you can manufacture products with consistent quality. Some of them are derivatives of natural compounds. So you look at the molecule from the natural source, and then you synthetically make something that looks like it. Or more recently, more of these novel adjuvants are actually drug-like compounds. So how do we find those? A lot of our contractors are using high throughput screening methods where you screen drug libraries for compounds that in vitro stimulate certain pathways that we think are associated with adjuvanticity. Now, more and more of our groups are switching to in silico screening. So you in silico screen actual existing libraries of compounds or hypothetical compounds that are computer designed, and they're only being synthesized if the algorithm predicts a sufficiently high likelihood of binding to the receptor and acting as an agonist and having the potential to be an adjuvant. Now, from our program, these are the numbers from our adjuvant discovery program. Our contractors so far have screened in vitro over 3 million compounds. At the same time, or actually in less time, fewer groups who have done the in silico screening have already completed the screen of more than 10 to the 24th compounds. Since this screening is really just dependent on how much CPU time you have access to, that's the only limitation. Now, why would you do that in silico? You can create focused compound libraries in silico, which allows you to reduce the size of your in vitro screening or the extent of your in vitro screening, the libraries that you need to screen subsequently in vivo. So that means it accelerates adjuvant research, it reduces the costs, and it reduces the risk of the project. And I wanted to show you at least one example of such a drug-like adjuvant. You may be familiar with the NPL adjuvant. NPL is a derivative of a bacterial endotoxin, the lipopolysaccharide. This has been known about this molecule for quite a while. This was discovered in the 1970s. It's a kind of it's usually called a detoxified version of the endotoxin. And it's a biologic. It's still being produced in bacteria, isolated, chemically modified. It's used in a number of vaccines. For example, the shingles vaccine, the Shingrix, which is approved in the US. The HPV vaccine is approved in the US. The malaria vaccine was approved in Europe or used in Africa. All of those use the NPL adjuvant. And it is structurally related to its parent molecule, the endotoxin, which is chemically modified. Now, another adjuvant, one that came out of our program, is called FOS47. So this is a combination adjuvant. It uses two different types of molecules stimulating two very different receptors. One is toll-like receptor 4. The other one is toll-like receptor 7 and 8. Now, what's unique about this combination adjuvant is that both components are small molecules. They have nothing to do with the naturally occurring agonists for either one of those receptors. When you look at the TLR4 agonist, it had absolutely no structural relationship to this to the natural agonist or its derivative like LPS. So here's that drug-like adjuvant molecule, which of course is much easier and cheaper to make, to formulate, because you're not dealing with this large molecule with a long carbon chain. Why is it even working? Because it interacts with the receptor through the correct side chains. So once you know what the interactions of that molecule in the relevant binding pocket of the receptor are, you can now start modeling the binding, and you can have the computer design drug-like molecules that have a high likelihood of mimicking the effect of the naturally occurring adjuvant. This combination adjuvant is currently in its first clinical trial. Now, a program has been in existence since 2003. A number of compounds have come out of it. But one of the criticisms we faced was that information about these adjuvants was difficult to find. So we published a strategic plan in 2019, which included a call for a database to make the information more easily accessible to the vaccine research community. And I usually refer to it as our dating website for vaccine researchers and adjuvant researchers, to bring them together and show the vaccine research community what is available and how do these adjuvants that are available actually work. The database was launched in earlier this year, excuse me. And here's what it looks like. The URL is very straightforward. It's a vaccine adjuvant compendium at NIAID.nih.gov. It allows you to search for adjuvants based on certain characteristics, what receptor it is binding to, what type of immune profile is induced. And that's important for vaccine developers who are looking for compounds that, that stimulate immune responses associated or presumed to be associated with protection against certain diseases. You can look at the stage of development if you want to de-risk your program. You can also search for adjuvants based on what pathogens, what disease models have they already been used in, again, as a way of de-risking your project. It's just a couple examples. This is what it looks like when you're searching for adjuvants. And these symbols here refer to the category that they're in. And I want to get back to this aspect, because this categorization is, of course, of interest from an ontology standpoint. The output format from the database is either individual data sheets, where you can see everything. This is just cut off, but it's the top of one of those data sheets. But it shows you all the available information about a particular adjuvant, all the publications, routes of immunizations that it is suitable for, clinical trials that have been conducted with it, and so on and so on. And most importantly, how do you get access to it? Who do you contact? Who do you talk to in order to start a collaboration? Which is really the main difference to any other adjuvant database out there, which lists adjuvants that, such as those in commercially available vaccines, licensed vaccines, that you will not have access to. Or, of course, you can export all the data from the database and then do your own searches. Now, the reason I'm talking about this in this forum here is we've recently linked the NIAID adjuvant database to a vaccine ontology database. And the objective here was, first of all, to put the adjuvants in the compendium in context. We want to harmonize the categorization of vaccine adjuvants, and this categorization of adjuvants continues to be a really highly contentious issue. Depending on who you talk to, in the field, you will get different categorizations. Everybody uses different criteria to categorize their adjuvants, which makes it really difficult to compare. Some of these categorizations are based on biological effects, others on origin, others on the chemical nature. We also wanted to widen the platform and the online presence of these adjuvants. And again, not just all adjuvants out there, but adjuvants that are actually accessible, that the vaccine research community would be able to work with. This was accomplished through a collaboration with Dr. Hayes' group at the University of Michigan, and we've started linking the records in the VAC to the corresponding records in his database in OntoB. Here's what it looks like. This is the adjuvant I just mentioned before. This is combination adjuvants, FOS47. This is the record. This is the title to the record in our database. And here is the linked record in OntoB. Now, going forward, one of the projects we've started working on is to assign vaccine ontology ID numbers to the adjuvants. And why was that necessary? What triggered this? Now, as adjuvant developers are moving through the discovery, through the development stage of their adjuvant, you often see changes. You see them changing the name of the adjuvant. You may start with some kind of a code, and then eventually, as it becomes a lead compound, it gets a proper name. And that can really confuse the readers of papers who are now seeing different terminology, different names. Sometimes PIs even use different names for the same adjuvants in different papers published around the same time. Another issue is if you formulate an adjuvant differently, or if you put it into a combination, such as the one I just showed you, FOS47, it is really a different product. The biology changes, the immune profile changes. So in other words, it is a completely different adjuvant, even if the core molecule is still the same. But if you don't give it a separate, unique name, once again, you're confusing the end users. And then, unfortunately, the investigators in the field, they don't necessarily talk to each other when they name their adjuvant. So you sometimes end up with very similar names. And my favorite example is an adjuvant that comes from our portfolio. If I have time, I'll show you a little more about this. It's called Advax. As a commercially available adjuvant for preclinical studies, it's called Advax. The two have nothing to do with each other. They're chemically, functionally, they're very, very different, but they're frequently confused with each other. So the solution was that OntoB assigns unique ID numbers to each of the adjuvants in our database, and that ID number links the record in for each adjuvant in the two databases. And what we're doing now, as the numbers are being assigned, the PIs, the IP holders of the adjuvants are being contacted, and they're being encouraged to use those numbers in publications. So that will make it easier for readers to go back and look up all the characteristics of the adjuvant mentioned in a publication. It also helps the investigators who don't have to provide as much background about the specific adjuvant anymore, because they can simply refer to this link. And the hope is that this becomes kind of a one-stop shop for looking up information about adjuvants. So this is what it looks like in the VAC, in the NIAID database. Here is the vaccine ontology identifier. It's a hyperlink that then takes the user to OntoB. Now, I do want to use the last couple minutes to talk about, just to bring out two of our stars in the program, two adjuvants that are quite relevant in the context of the current pandemic. The first one is Alhydroxiquim-II. And the lesson here is that it is possible to go from a very early stage adjuvant very early stage product to an emergency use authorization in less than a year. If you had asked me 2 years ago, I would have said that's absolutely impossible. The second one is one I just mentioned, Advax, but in a combination adjuvant together with a toll-like 9 receptor, a CpG oligonucleotide. The lesson here is that an adjuvant can actually be anti-inflammatory, which again, if you had asked people in the field a couple of years ago, they would have told you that's just not possible. That's counterintuitive. And it is also a success story of using in silico screening to identify novel adjuvants. Let me just briefly give you some background about Alhydroxiquim-II. Alhydroxiquim-II is a TLR7 and 8 agonist. So it targets TLR7 and tolerant receptor 8. Those molecules are not per se new. They've been used for a while. They've previously been approved as topical treatment for cancer, for warts. However, so far they've only been used as topical therapeutic agents. The reason is that they are quite potent. They do have significant side effects. These include local inflammation, blistering, burning, skin redness, dry skin, skin breakdown, crusting, scabbing, but also systemic effects such as fever, flu-like symptoms, headache, tiredness. So the problem is that these are potent, but they can be too potent. So how do you contain the side effects, or how do you eliminate the side effects without affecting adjuvanticity? In case of Alhydroxiquim-II, the molecule, the TLR7 agonist, the small drug-like molecule, is attached to alum. And in this way, you are delivering the active molecule, the TLR7/8 agonist, directly to the lymph node, which means you don't get what's being called in the field wasted inflammation, an inflammatory response that doesn't contribute to adjuvanticity. By using that targeted delivery approach, you're also getting significant dose sparing because you don't need as much of the molecule to get enough of it to the draining lymph node. So it's dose sparing of both the antigen, because you're using a potent adjuvant, and the adjuvant itself. By attaching a TLR7/8 agonist to alum, you're also changing the immune profile of the response. Alum, as I mentioned before, induces a Th2 type immune response, useful for certain things, but not for many other vaccines. And attaching a TLR7/8 agonist now changes the immune response to Th1. How is this accomplished? Here's a very high-level summary of the chemistry. Here's the core structure of aluminum hydroxide, and the small molecule TLR7/8 agonist is attached using a chemistry that's coming from nature. This is the same kind of linker, or the same kind of chemistry that mussels in the ocean use to attach themselves to wet rock. There's a very strong bond, and however, the small molecule TLR7/8 agonist does come off in the draining lymph node in order to be able to bind to its receptor. Now, this molecule was developed through our discovery program. It's currently being developed as an adjuvant for a dengue vaccine, but also for a subunit COVID vaccine. However, in addition, it was licensed to Bharat Biotech in 2019. Initially, it was meant for Bharat's dengue vaccine. When the pandemic started, it was also applied to their COVID vaccine. This vaccine is known as Covaxin. The trial started in the summer of 2020. And keep in mind, up to that point, this adjuvant had not been in people. The EUA in India was issued in January of this year. The The vaccine is currently used under an EUA in 14 countries, pending EUA approval in several dozen other countries. It's been licensed to companies in North America, not yet approved, but those companies are working on it. To date, it's been used in this vaccine, and therefore this novel adjuvant, which a little more than a year ago had not been in a single person, has now been used in more than 30 million people with very, very few side effects, no serious side effects. It's a very safe, very effective vaccine. Why is it such a big deal? Now, with this first clinical use of Alhydroxiquim, this has a number of implications. Now, from our standpoint, it's a great success story because it was the first product from our portfolio that made it into a globally used product. What's also amazing is the development time. From the time it was discovered to approval, it was 2 to 3 years. Now, compare this with the adjuvant I mentioned before, monophospholipid A, MPL, which is part of 3 approved vaccines. That adjuvant was developed in the 1970s. The vaccine, the first vaccine, including MPL as an adjuvant, was Cervarix, the human papillomavirus vaccine. That was approved by the FDA in 2009. Now, it is the first TLR7/8 agonist in any infectious disease vaccine. As I said, it's been used before for topical applications, but not for vaccines. And that means it will lower the barrier for other TLR7/8 agonists as adjuvants. What's also really unusual is this is a product that comes from a very small biotech company in Kansas. This is not from a big pharma company like all the other adjuvants that are in the commercial product. And there are competing products, TLR7/8 agonists, conjugated to alum from GlaxoSmithKline, from 3M, that had been under development for a longer period of time, but are not in product yet. It's also the first approved COVID vaccine that globally that's based on a novel adjuvant. And the next one we expect to be approved is the one from Novavax, and that uses a very different type of adjuvant that uses a sapropin-based molecule. Now, and I just wanted to point out this is where TLR7/8 agonists work in cells. Now, the second adjuvant I wanted to highlight that comes from our portfolio for a number of reasons. I picked this adjuvant is Advax. And what's unexpected about this particular adjuvant is it's a sugar. So how do you use a sugar as an adjuvant? Now, inulin in its natural form is truly a global commodity. It's a fructose polymer. And you may actually have had some for breakfast this morning. It's in yogurt. It is in a lot of food products. It is used by plants as a storage molecule for energy, and most of the commercially available inulin comes from chicory roots. There is a medical application. It is used to measure kidney function and excretion of this molecule, but in its water-soluble form. How do you turn this into an adjuvant? If you formulate it into water-insoluble crystals, and then rather than ingesting it, you inject it, it acts as an adjuvant through still somewhat unclear mechanisms and receptors. This type of adjuvant has been used by now in more than a dozen clinical trials, countless preclinical studies for pretty much every disease indication. What's also unique about this adjuvant is it challenges one of the old dogmas in the field, and that is, in order to get an adjuvant effect, you have to induce an inflammatory response. And not only does this adjuvant not induce inflammation, it suppresses inflammatory responses. And one of the clinical trials actually showed that when formulated with a commercially available licensed flu vaccine, it reduces the number of adverse events, because usually you would expect an increase in adverse events once you add an adjuvant. Now, the current version of this adjuvant, it was used as a pure sugar crystal before, but the current version is a combination adjuvant with a CpG oligodinnucleotide. So these are short DNA oligos that are synthetically made. They bind to one of the other toll-like receptors, TLR9. A CpG oligonucleotide was approved in the context of a hepatitis B vaccine, HEPLISAV-B, from Dynavax a few years ago. Most of the CpG oligodinnucleotides that are out there are based on naturally occurring bacterial DNA sequences that turned out to stimulate the human immune responses. Now, the CpG adjuvants are potent adjuvants, but they can be pretty reactogenic. And another major shortcoming is they tend to be species specific. So whatever you test in mice, whatever responses you get in mice may not translate to what you're going to get in humans. A few years ago, we started supporting an in silico screen for novel CpG sequences. These are computer-generated sequences. These are computer-predicted sequences, what is being measured here is the predicted binding affinity to TLR9 once the sequence is identified as having a potential ability to bind tightly to TLR9. The DNA oligo is synthesized and then tested in vitro. So these are non-natural sequences. They turn out to be much more potent than those that naturally occur. They are also multi-species specific. So you can now test the same molecule in mice, in rabbits, in ferrets, and then translate those findings to humans. Here's the lead sequence, the lead oligo from this screen. It's been in several clinical trials already in the US and Australia. When combined with Advax, it turns out that the two adjuvants, chemically very different molecules, likely targeting very, very different receptors, they turn out to be inducing very synergistic adjuvanticity without reactogenicity. You get adjuvant dose sparing, which means you can use much less of that CpG oligodinnucleotide. And one interesting feature is that this combination works in newborn mice. CpG alone does not. So this is a potential adjuvant for childhood vaccines, for early childhood vaccines. Why did I pick this? I mentioned some of the reasons why I highlighted this adjuvant, but another reason is that it is used in a COVID vaccine project where we started supporting the preclinical evaluation. This was last year in March. It was tested in mice and ferrets, non-human primates, and it turns out to protect even old non-human primates, which a good model of senescence and of mimicking frail elderly humans, which are hard to immunize and protect against most infectious diseases. But this adjuvant induced very strong immune responses even in those old non-human primates. The vaccine is currently being tested. Well, the testing started in Australia in phase 1. These were completed at the end of last year, and it is currently in phase 3 trials, and we expect data later this year. And if everything looks good, which it does, it seems to be quite effective against the circulating variants. Hopefully, this will be one of the next COVID vaccines to be approved early next year. Thank you very much, and I'd be happy to take any questions you may have.
Great. Thank you very much for your wonderful, inspiring talk. So to introduce vaccine concept and a lot of wonderful stories, right? Like the first kind of adjuvant, which has been put in use, approved in less than a year. Wow, and then used for COVID. So that's a wonderful story. And the other one, the Advax CpG from plant, right? And it's a CpG. Also, it's a wonderful story. I'm an immunologist myself in training, so I was studying CpG for a long time. So but now it seems like there's a lot of going on there. So that's a wonderful talk, and also working with Wolfgang by having the vaccine adjuvant standardized using vaccine ontology. I think that it's a brilliant idea. Thank you for seeking us for collaboration. Yeah, very wonderful talk. Okay, now let's see if we have any questions. Like Asia.
Yeah, hi, this is Asia. Sorry, do you hear any echo? No?
Fine now. Not anymore.
Okay. Oh, good. So actually, I'm very happy you are appearing here. I just sent you an email. I was Oliver's postdoc, and before I go to FDA, so I just joined NIH last year in December as a data scholar. So I have been working with Oliver. I still work with Oliver on various ontology projects, but my thinking is that now as you collaborate with Oliver, I want to make a connection with FDA. So the adjuvant story is really interesting. This is a question for you. Like, I was struck by only 5 novel adjuvants being approved by FDA. So I don't think FDA approves adjuvant separately.
No.
Okay. Okay.
These are, you're absolutely right. Adjuvants are not approved alone unless you're proposing to use them as standalone immunotherapeutics. They're only approved in the context of a vaccine. So when I say approved adjuvants, it is approved vaccines that contain novel adjuvants.
Right. Mm-hmm. Yeah, so maybe there is no regulation to just approve adjuvant as itself. But I'm wondering.
And there's a good reason. There's a very good reason for doing that, because if you combine one adjuvant with 2 different vaccines, the profiles are going to be different because the antigen itself changes the immune response, it changes adverse events. So right. it makes sense to not approve the adjuvant by itself.
That's correct. Yeah, there's no intended use. So what I'm seeing is that do you have so when you show your figure, you said your adjuvant will support different in NIAID groups as well as developers. Do you have an arrow point out to like the users such as important user as FDA?
So we do actually have ongoing research projects with FDA investigators who are testing adjuvants from our program in FDA labs. But this is different from interacting with the regulators. We don't get involved in the discussions that the IP holders of our adjuvants have with the regulators at the FDA.
Yeah, right. What I meant, sorry.
No, go ahead.
Yeah, what I meant is more like informatics level, like the data that the resources that NIH supported or Oliver developed should be leveraged by the FDA CBER. I think I can make that connection. So I hope that can be continued after the this workshop.
Absolutely. Yep. Let's talk offline.
Right. Thank you.
Yeah, that's great. Thank you. So
yeah, I have one.
Okay, go ahead.
It's more in a comment than not. the question, and I think that is really great what you have been presenting. And, I think is a great resource, the fact that you are linked to the vaccine ontology. And the other thing that I see as I'm envisioning, continuing going with this approach is the fact that not only the link to the vaccine ontology ID will be provided, but it could have been implemented by the description of the mechanism of the various, adjuvant associated with the specific antigen in the vaccine. And that could kind of be really helpful in then understanding the role of the adjuvant in a specific context, and be able to even clarify when an adjuvant will be most active, to give an antibody response against a cytotoxic response, or things like that. So I really like the idea of linking to the ontology, and I'm really envisioning the expansion of that through the vaccine ontology and other ontology. So.
Now you mentioned the description of the mechanism of action, and we purposely decided not to use mechanism of action as a criterion, because for a lot of the adjuvants, the mechanism of action is unknown. We don't know the receptor. Think about Advax. I mean, used in more than a dozen clinical trials already, very safe. Every study to determine a mechanism of action, I wouldn't say has failed, because the outcome was that this particular receptor is not the one that Advax is working through. So we learned something. What we don't know is what's the right mechanism. So for a lot of them, we just don't know, and therefore it's not a useful readout. We are, just wanted to mention, we do have a large program right now. It's solicitation just closed last week to specifically characterize mechanisms of action of adjuvants in standardized models. So use the same vaccine, use the same readout for panels of adjuvants, because one of the shortcomings of in the field is, if you are taking the data from different papers from different studies, everybody uses a different readout. Everybody uses a different in vitro readout, in vivo readout, and it's really hard to compare even immune profiles between adjuvants. So this will hopefully also help kind of standardize the characterization of adjuvants.
Yeah.
But more about that in a few years.
No, I agree. And I know that sometimes, the most of the time you don't know, the mechanism is not known. But for example, knowing in your the vaccine where you use Advax and CpG, then you know which are the two receptors working in one case and the other, and so you could kind of capture at least that, and that will help knowing that you are going through one node receptor and a TLR receptor together, and they can synergize through the response. Because looking at other system, I'm saying, so you could kind of take advantage of what other system have already known and put it linked to the vaccine and the adjuvant. I think will be really important to start at least. It could.
Yeah, I think I can add something here. So I agree with Wolfgang. We cannot use it for a big high-level thing, but for some, I also agree with Anna Maria, right? For some, we do have, right? Like as Wolfgang showed, like some adjuvant do have some mechanisms, right? Like receptor 7, 8 agonist. So we have indeed, when I present, I was modeling, representing the adjuvant from Wolfgang's work. When I know the mechanism, I was trying to model it actually in vaccine ontology. So we can do some of them. For now, I think I put it as a row, right? Like adjuvant row, right? So you have the total receptor 7, like agonist low, something like this. Yeah, we can do some of it. Yeah.
Yeah, and I have done 2 years ago in collaboration with Lindsay and Alan Rutenberg, a TLR signaling ontology that I think could be revise a link to that and kind of, put in, that is a formal representation of the signaling. So it's been used in part from Reactoma, but there is all there. And I think that could be, one way where you can put things together, knowing from what you know, obviously you cannot put things that you don't know. But I was very intrigued by the Advax and the CpG because I see from my immunological background that, you are talking about two synergic mechanisms coming from two different receptors, and would be nice to kind of formalize that and capture.
Yeah, it's good.
Couldn't agree more.
Okay, so yeah, it's very good talk and a very good discussion. Possibly due to the time limitation, maybe we can end the keynote session, and then we can go for next round. But definitely we can keep like a back end, right? Like or after talk, after the workshop, we can keep communicating. Yeah, if you have any ideas or questions, or you can put your question into the chat, and we can continue to have some discussion. Great, thank you very much, Wolfgang, for your wonderful talk.